at Glasgow, and then he went on to do a PhD in molecular and cell biology at UCL Manchester, and then he moved on to do two postdoctoral positions in Edinburgh, firstly at the MRC Human Genetics Unit, and then later at the Veterinary School in the University of Edinburgh. Alasdair Mackenzie joined the University of Aberdeen in 2001 and has remained a principal investigator ever since. The Mackenzie Lab at the University of Aberdeen investigates enhancers, and some of you may know that enhancers are cis-regulatory elements in the sense that they regulate there are regulated elements present in the human genome which have important roles in gene expression regulation. And the lab is particularly interested in how we can regulate gene expression and understand the gene expression of obesity, anxiety, and alcohol abuse genes. The Mackenzie Lab has a recent breakthrough using CRISPR-Cas9, which is the genome editing tool which won the Nobel Prize for Chemistry in 2020. And that was used to make selective knockouts of enhancer regions in mice models, only in the span of a few weeks, and that was quite remarkable. So I know from my experience working with these cells and string constructions and mouse knockouts, it's quite a lengthy process to do from what I know. And basically, in short, the CRISPR deletion knockouts showed a decrease in mice fat and alcohol intake, and I'm very sure Prof Mackenzie is going to talk about that in his talk today. And this is important because understanding genetic differences, it contributes to the advent of personalized medicine, because we have seen for the COVID-19 pandemic and other treatments nowadays is that one size doesn't really fits all when it comes to medicine. That's why nowadays personalized medicine is a very big thing. So without further ado, I will send over to Professor Mackenzie to talk about his talk today about CRISPR-Cas9 in the context of alcohol, obesity, and anxiety abuse. Over to you, Alasdair. Thank you.
Thank you, Alasdair, for that introduction. I suppose the first thing I have to say is that I'm not a bioinformatician or a biologist, so I'm most interested in the basic biology of gene regulation, specifically in how the non-coding genome regulates the expression of genes and how that can go wrong in disease. And so why is this important? Oh, here we go. Okay, then, so are you going to progress? Okay, I'll just say click. I'll say click if it doesn't work. No, it doesn't work. Oh, it does work. Okay, so you're all familiar with genome-wide association studies, whereby thousands of people, thousands of patients and unaffected cohorts are studied for changes in their genome sequence that can be associated with disease susceptibility. And so over the last 20 years, there's been about 5,000 to 6,000 GWASs carried out on diseases ranging from heart disease to alcoholism and all the way to even height has been looked at from GWAS. And so a study of all these GWASs, when they took all these GWASs together, what they came up with was this very inconsiderate figure of between 90 and 95% of the most associated loci were outside of coding regions. Now, because most biologists set up to study coding regions, this caused a bit of a problem. Also, another study which suggests that the majority of SNPs in the genome could actually working together cause most complex disease. They looked at where the majority of these SNPs lay in the genome, and their conclusion was that the vast majority of these SNPs which cause complex disease in humans tend to lie in regions of the genome which control cell-specific gene expression. Now, why is this important? If we look at the human body, the first thing you notice is that we're multicellular. That's a really important aspect of our biology. We can't ignore it. I think a lot of biology does ignore it. It tends to think in terms of single cells or proteins, when as a matter of fact, we've spent the last 700 to 800 million years of our evolutionary biology developing as multicellular organisms. So we can't ignore this when we start to look at the causes of disease. So in multicellular organisms, it's important to realize that genes have to be expressed in the proper cells, in the proper amounts. And this is another aspect that we need to get to grips with in response to appropriate cues. So for example, if you suffer an autoimmune disease, there's a good chance that what you're seeing in an autoimmune disease is the fact that these genes are expressed where they shouldn't be. So they're responding, whatever's controlling these genes is expressing inappropriately in that condition. So we cannot ignore the fact, and this is the take home, if there's one message you take away today, it's that we have to consider health in terms of a multicellular organism where cells interact to maintain health, and when these communications break down, leads to disease. So if there's one message to take away today, that's it. Okay, then again, there we go. So I mentioned cell-specific regulatory networks, so I'll just describe to you briefly what these are. These are systems within the cell. So this is a cell here, just like the plasma, and when you touch the screen and so on. If you look at a cell, the actual mechanisms within a cell that allow cells to communicate, one of the most important components is cell-cell interaction, where ligands and receptors interact at the cell surface. This triggers signal transduction cascades, which are excellent, which are being used a lot for the development of drug targets. And these signal transduction pathways terminate in the activation of transcription factors, which bind to the genome. And where these transcription factors bind, there's a series of different classes of regulatory element within the genome. And the one I'm most interested in for this presentation. So here's how it works. You get transcription regulated by activated transcription factor binding to enhancers in the gene. Okay, so altogether, these are known as cis-regulatory elements, but the ones to talk most about are enhancers. Yeah, here we go. Yep. Okay, then. Bit of a delay when I press the button, it seems to not move. Okay, so what are enhancers? I'll give you a brief view of the biology. They're sequences of DNA. They can lie anywhere within a megabase of the genes they regulate. They're only 200 to 500 base pairs long. And active enhancers, I won't go into a huge amount about the biology. This is just a cartoon of the accepted dogma surrounding enhancers in terms of how you can identify them. And one of the ways that most people use are these markers here, which are histone methylation markers. And so the histones associated with the DNA around these enhancers, when the enhancer are active, have these specific, which we can detect using antibody assays. Unfortunately, right. Okay. However, enhancers are not active in all cell types, and this is actually the basis of a phenomenon. Which again, if you take away a message, it's a phenomenon called context specificity, which in the past has been called cell and tissue specificity. And the tissue and cell specificity of gene expression is controlled by the context specificity of these enhancers. This kind of complicates matters because the majority of enhancers which are important to health are not active in cell lines. And so they're really hard to detect in cell lines because they're not active. So that is a bit of an issue. And what I hope to talk to you for the rest of this talk is how we put around that problem thanks to that big brown building next to it. Okay then, so one of the things we realized earliest 10, 20 years ago was that if you look at the expression of genes in different higher vertebrates, what you'll find is that genes are expressed in very similar ways, in very similar patterns, in similar regions of the brain. So across the whole of evolution, for example, here's a pituitary, and we see many of the genes expressed in pituitary, and what we can see is that in the majority of cases, we can see that the same genes are expressed in the same tissues and in the same cells in all of these vertebrates. So the question was, has the information required to support the tissue-specific, context-specific expression of these genes being conserved as well? And what we did was we decided to examine this in the context of alcohol abuse. Just give you a couple of a little background about alcohol abuse. As you know, whiskey's very Moorish, but causes 5.3% of all global deaths, and in some countries, up to 10% of male mortality is caused directly by alcohol. I'll just keep pressing this until something happens. Also, a comorbidity of alcohol abuse are anxiety disorders. And again, this has a lifetime prevalence of up to 32%. There are 264 million sufferers worldwide. And one of the major problems is that 1/3 of anxieties are treatment refractory. In other words, they don't respond to classical treatments. And these are very often comorbid. So if you have alcohol abuse, quite often it's because you're medicating yourself to reduce the anxiety. But many people, when they drink a lot, they get more anxious. So there's a sort of feedback, positive feedback between these. So one of the genes we're interested in is one called GAL, the GAL gene. It expresses a neuropeptide called galanin. And galanin is expressed in, it lacks coding. It lacks, this coding sequence of the galanin gene lacks SNPs. So we asked the question, what controls the expression of the galanin gene, which is very discretely expressed within regions of the hypothalamus and also regions of the amygdala. And so where it's expressed in the hypothalamus seems to be involved in controlling alcohol intake, and in the limbic system, or the amygdala of the limbic system, seems to be involved in controlling mood and anxiety. So if you use drugs to block the receptors of galanin, you can modulate mood and alcohol intake in mouse models. Let's keep pressing. There we go. So if we look most closely at the hypothalamus here, and if we make a map of the hypothalamus, the specific clusters of cells in the hypothalamus, we can see that galanin is a neuropeptide expressed in only specific groups of cells in the hypothalamus. These cells control things like appetite, alcohol intake, and so forth. So what actually controls the expression in these specific cells? And it's interesting that expression of the GAL gene in these regions of the brain have been conserved almost perfectly between mice and humans. So. Okay, so we asked, can we identify the regions in the genome which control the expression of galanin in these specific regions of the brain? And the first thing we did was, this was in 2005, between 2005 and 2010, we got access to about 100 different genome sequences of higher vertebrates, all the way back to lizards. And what we're talking about here, when we come to lizards, we're talking about 400 million years of divergent evolution. And the first thing you'll notice is that the galanin gene, the exons of the galanin gene have been highly conserved, no surprise. But there's also a lot of conservation outside of the galanin gene. That's what we're interested in. So what we did was we identified this piece of DNA, which we call GAL 5.1. It lay 42 kilobases away from the galanin gene. And the first thing we did, across in the big brown building there, worth mentioning, the poster I showed you beforehand, where you showed all of the methylation signatures, this sequence showed no methylation signature. If you've heard of the ENCODE consortium, the ENCODE consortium said that all of the enhancers in the human genome can be identified thanks to these methylation signatures. Unfortunately, we didn't see any of them in our enhancer, but what we did was across the big brown building, we tested its activity as an enhancer. So we linked it up to a gene that goes blue, and we microinjected that into mouse embryos. And here's the expression of mRNA within the hypothalamus of a mouse, showing where the galanin gene is expressed. Lo and behold, when you take this piece of DNA and you link it to lactate, it drives expression in exactly the same cells. Okay, so we also noticed that in the human population, this enhancer region, we can now call it an enhancer region, contained 2 polymorphisms, SNPs, and we then carried out a series of assays in cells to see whether these changed the activity of the enhancer. And what we found was that yes, there was a strong influence, depending on the allelic variation within these SNPs. There was a strong correlation with changes in its activity. So then we paired up with some people in Edinburgh, and they, using the UK Biobank, I won't go into the details, they used human data to show that there was actually a very strong correlation between male alcohol intake and anxiety associated with one of the allelic variants of this SNP. And that also ties back to a series of studies done in the late 90s, showing that there was a correlation between SNPs and anxiety in certain populations. So we were happy that there's something happening here in humans as well, which seems to mirror what we're seeing, what we hope to see in mice. So what we did then was, this gave us the excuse to jump aboard the CRISPR bandwagon. And so we designed CRISPR guides to knock this out in mice.
Well,
I think there's just a delay rather than. So we use CRISPR to knock it out in mice across the big brown building, where we've got a state-of-the-art genome manipulation suite. And then Andy, who's sitting right there, decided, well, thanks to a grant, he then looked at the expression of galanin in these mice. And what we find, here's the galanin gene here. This little gene here. Here is where our locus, our enhancer is. So first of all, to be thorough, just to do due diligence, you look at the expression of, so here's the galanin gene, the mRNA of the galanin gene. And what we also see is where this piece of DNA drives expression of a reporter gene within mice. But if you look at the expression of genes surrounding the galanin locus, they were unaffected by the knockout. However, when we looked at the galanin gene itself, it was practically switched off in the hypothalamus, and it was also switched off in many of the other components of the brain, including the amygdala and the hypothalamus. So then that triggered a series of questions regarding what were the effects of knocking this enhancer out on the behavior of the animal. And the first thing we did was we looked to see what its effects were on the intake of fat, because we know that galanin has an effective fat intake. And we showed that animals lacking this enhancer took in significantly less fat. But what was most exciting was that in addition to the fat intake, they also didn't like ethanol very much either. So this led us on to looking at the anxiety phenotype of these animals. And we did a series of tests. I'll only go through 2 of them. So we can see expression of galanin in the medial amygdala. And we can see that the enhancer supports expression in these regions as well. So we carried out a series of anxiety tests on these animals. And the first thing we found was using a thing called the elevated zero maze, where the animals either hide or they stay to the open. We can time the times they're in, either closed part of the maze or in the open. And that tells us about the mental state in terms of anxiety. And what we found was that, mirroring what we saw in humans, the males seem to show the highest reduction in anxiety if you knock out the enhancer. And we repeated this in another test, which a lot of people enjoy hearing about, which is called the marble burying test, whereby animals which are less anxious, they bury more marbles. It's just a behavioral quirk of mice. And Joe here was able to show that in mice lacking the enhancer, there was considerably less anxiety in these animals as well, in both sexes. And we then went on to, so here we can see quite a significant reduction in both sexes of anxiety in these animals. Then we went on to ask, can we identify some of the transcription factors which bind these enhancers? I won't go into much detail because I'd like to get going through the talk. But we were able to identify a transcription factor which bound the enhancer and which seemed to be involved in this activity. I think for those of you interested, I can answer more questions about this. So I think this shows quite reasonably persuasively that these enhancers are highly conserved. And because they're highly conserved, this allows us to model human enhancer activity in mice. So then we get to finally talking about how we're doing for time. Oh, nearly there. We'll talk a little bit about epigenetics. So in addition to looking at the differences in polymorphisms, we were also able to explore the methylation of this enhancer. And we were able to show that if you took pups and exposed the pups in utero to high levels of fat, that significantly changed the distribution of methylation within the enhancer. And we know that methylation is a major epigenetic component. And in addition, I'm pressing the button as hard as I can. So we were able to show there was a significant change in the methylation profile of this enhancer as a result of being exposed to high-fat diet in the womb. And what we were also able, yes, probably a battery, the battery's running out in this. Next time. And we were also able to show it at the level of individual CPGs that high-fat diet significantly changed the methylation profile in specific and often highly conserved CPGs within the mouse and human enhancers. Okay, so that's as far as I'll take this part of the talk. I'll just finish up with a little bit. Yes, this also shows that methylation seriously affects the activity of the enhancer. Okay, then, so now we'll talk about how we can take this information forward and start to ask questions about the rest of the genome. And in this case, we were interested in SNPs, GWAS SNPs, which occurred within these enhancer regions. And we turned to a different gene, the BDNF gene, which we now know is involved in controlling things like substance abuse, fat intake, and anxiety as well. And so by overlaying these different databases, GWAS conservation, the UK Biobank data, and also expression quantitative fleet loci, which I couldn't go into in more detail for those of you who are interested, we were able to identify candidate enhancers within the BDNF locus. And specifically, a highly conserved region within the BDNF gene itself. Yeah, so here's a region of conservation, and we can see the SNP associated with obesity sits squarely right in the middle of this region, which had been conserved back to amphibians. We also showed that there was a change If you change the allelic, the allele of this SNP, change the activity of the enhancer. And so the first thing we did was we made a knockout line of this enhancer and hoping to see an obesity phenotype, hoping to see a change in either gene expression in the hypothalamus or its intake of food, or the intake of food or weight gain in the mice. And although we got a slightly significant result, it wasn't huge. We were slightly disappointed by that. But what then happened was we started getting feedback from some of the amazing people across in the big brown building that our mice seemed to be more anxious. They were jumping out of the cage. So we looked at the anxiety phenotype, specifically gene expression. So we gene expression within the amygdala instead of the hypothalamus. We looked at the amygdala because thanks to Felix Grassmann, who's now moved back home to Germany, he interrogated some human databases to show that there was a significant correlation between this SNP and anxiety in human cohorts. And so we looked at the anxiety phenotype in mice. We were able to show, first of all, that the knockout caused a major change in the expression of BDNF in the amygdala. And then when we carried out behavioral analysis of these animals, we showed that the females lacking the enhancer showed increased anxiety. And we could also reverse this anxiety by giving the animals diazepam. Okay, so I think I'll leave it there. I think we're out of time, but that's just some conclusion slides. Okay, yes, but we showed that the EQTL analysis did something as well. So again, I can talk about, for those of you who are interested in EQTL analysis, I can talk a little bit more about this afterwards. Okay, so conclusions. So data mining is a rapid method of identifying context-specific enhancers, specifically conservation, GWAS, and EQTL. And the study of enhancers conserved between humans and mice allows us to model their biology in vivo. And focusing on conserved enhancers will also allow us to understand the genomic basis of ancient behaviors. I mean, we're talking about behaviors which are common to all higher vertebrates, which are also associated with most of the things that go wrong in us. And that includes anxiety, appetite, reward pathway. And it will also allow for an understanding of how genetics and epigenetics interact at the level of gene expression, thus contributing to conditions that will probably affect most of the people in this room. Okay, and I'll just thank everyone who were involved in this. It's funded by the BBSRC and Medical Research Scotland, and I'll be happy to take any questions.
I just wanted to say to the people who are joining us in Zoom, feel free to ask your question as well by writing your question in the chat window so we can pick up on it. We don't want to exclude you from asking any questions. So should you have any, just write them down and we'll be sure to push them forward here. Any questions? Yeah.
I might have missed it, but is the GAL gene, is it part of the GLP-1 pathway?
Oh, I think it's an overlap.
Just the symptoms, like just the effects on obesity, on anxiety, on
Yeah,
like,
well, it's a hugely complicated wheels within wheels type
I'm
of thing.
just trying to think Ozempic, can you have
It's similar to GLP. It's a neuropeptide system as well. So I think the GAL neuropeptide binds the different receptor, but I think it's also there is some evidence that it also has an effect on the pathway you mentioned as well. Yeah.
And is the next step to understand how you can therapeutically target?
No, I'm just interested in the biology. I love the biology. I mean, if we do, Andy, if you can step up the question if you like. I just love the biology of these things. Yeah. There is a therapeutic potential, absolutely. We'd love to explore it, but it's mainly the biology I'm interested in. And I'm happy to collaborate with anyone who thinks that this could be geared towards a therapeutic. But I think the important thing is that we understand what's happening here. Before you can actually have a therapy or make a new diagnostic or a new drug, you have to understand what it's doing in the genome. And because 95% of the SNPs which are most associated with disease are in the non-coding gene, we need to understand what's there. So, but yes, nice question. Yeah.
I also have a question though, because I've read before online there are non-coding RNAs that regulate the expression of obesity genes. So I was wondering, is there any non-coding RNAs that also regulate the expression of galanine, the galanine gene, for example? And
Yeah.
maybe non-coding RNAs could be used as a possible therapeutic for controlling alcohol and anxiety abuse. So.
The role of non-coding RNA is very complicated. So one of the things that I skipped over was the fact that an EQTL within the enhancer, which we looked at in the case of BDNF, didn't control the BDNF gene directly. If you listen to it, if you look at the EQTL data, it controls the expression of its antisense gene. So from that point of view, yes, non-coding RNA seems to be really important, and we've only just scratched the very surface of the top of the iceberg in terms of both its activity and its role in gene regulation. There's also people who are interested in things called enhancer DNA, enhancer RNA, which is very similar to long non-coding RNA, but that could also be I've read papers where these long coding RNAs are actually the byproduct of transcription from enhancers, bringing enhancers and promoters together. The product is basically long coding RNA, which is then degraded. But the activity of bringing them together seems to be driven by RNA polymerase, which pulls them together, and the smoke that comes out the top of the chimney is long coding RNA. So whether it actually plays a role in biology, we still need to drill down and find that.
They could like gene therapy be involved in like therapeutic for.
I think that there's a gene therapy with antisense RNA is a real possibility. Yes.
Interesting. Thank you.
Yeah.
We have Robert James sharing in Zoom. He doesn't have a biology background, but he finds it very interesting that people might be able to reduce their anxiety. So,
Yes, tell me about it then.
Yeah.
Yes, so as I said, 1/3 of people who suffer anxiety recurrently are refractive to current drugs. And yes, any way we can understand the mechanisms leading to anxiety will give us an idea of how to deal with it in the future. Also, if we can actually identify those who are more susceptible to anxiety, you're halfway to a treatment.
Yeah.
And there's also the whole personalized aspect as well. If we can identify those people whose enhancers might overregulate or underregulate the expression of anxiety-related genes, then we can identify these people and maybe not put them in positions where they're going to be subject to a lot of pressure.
Yeah.
Yeah. So yeah.
I have one question around CRISPR.
Yeah.
So, for better or for worse, I get very excited when you hear CRISPR and all the possibilities that it has to offer. Have you seen any downsides? Are there any kind of things like, well, hold on, this is going a little bit too fast, or this is too much, or it becomes a magic wand? We overpromise.
I think there's a lot of hype around it. I think the actual CRISPR process itself is incredibly simple. It's just you get a protein, you wrap it in you basically code the protein and tell it where to cut. That's what the entire technology is.
Yeah.
And so people then overhype it as, oh, this is going to be the way that we cure all human disease.
Yeah.
We're not even a tenth of the way there to be able to use CRISPR therapeutically in a safe manner.
Yeah.
So I think we got to understand what's hype and what isn't hype. But it's an amazing tool. I mean, I wouldn't be standing here if it wasn't for CRISPR technology and our ability to use it across in the big brown house.
Yeah.
And so we're actually gearing up to do another big CRISPR run in the next few weeks with the mice across there. So we've got a really nice transgenic unit over there, and we can I think, how long did it take us to produce the last one, the last mouse model?
Few weeks.
Well, it took us 3 hours to make it.
Yeah.
It took us 6 weeks to tell it it worked,
Yeah,
which was a frustrating 6 weeks.
and then another 3 to 4 months.
Yes.
Crossbreed the mice
Yeah,
back
so,
and
but the actual
then use
production of these CRISPR mice is incredible. quick.
knockouts. So,
You can do it in a morning. Whereas beforehand, using embryonic stem cell technology 10 years ago, it would take you over a year and cost you 100 grand. You can do it for 3 hours for a couple of grand over those. Very accessible technology.
Yeah.
But it can be overhyped.
Any more questions? Then thank you very much, Professor
you.
Mackenzie, for this great presentation. So this brings us to our